Sir, I'm working with neurons.
Okay.
1 minute.
Okay, YouTube is ready now. Nikita, you can start.
Good afternoon, everyone. So I, Dr. Nitika Thakur, on behalf of Microbiologist Society India, welcomes you all to the series of Indians talking from abroad. And today we have a very important lecture by Dr. Nagesh Kadam. So Dr. Nagesh Kadam is working as the postdoctoral fellow in Institut Curie, France. Dr. Kadam has worked under the kind supervision of Dr. Kavita Babu as a PhD research fellow in the Department of Biological Sciences, ISR, Mohali, India, on the topic, the study of reversal behavior and chemotaxis in Caenorhabditis elegans. He adds to his name many high-impact research publications, conferences, and is a recipient of many fellowships and awards. So he'll be talking on the topic C. elegans as a model system for study of various biological aspects. Over to you, sir.
1 minute. Dr. Patankar will talk for 2 minutes.
Okay, sir. Sanjeev,
Dr.
sir.
Patankar, please.
Over
Dr.
to you
Patankar.
first.
Nagesh Kadam, I think Nagesh has left. Probably, yes. Nagesh
Yes,
has left.
yes.
Patankar, sir, you
I just
can
contact.
talk. Yeah, he has joined. He has joined. I have made Nagesh as a co-host. Patankar, sir, please.
Hello, sir.
Yes.
Sorry, actually I was disconnected. I don't know.
Yeah. I have made you co-host. You can try for your PPT presentation. And
Okay.
Dr. Patankar, please.
Hello.
You start. You give in 2 minutes. You give introduction of Microbiology Society.
Okay. Hello. Am I
Yes.
audible?
Yes,
Yeah,
sir.
yeah.
Yes, sir.
Good afternoon to all of you. I welcome you all in this lecture series, 21st lecture, Indian speaking from abroad. I am Dr. Sanjeev Patankar, National Coordinator, Microbiology Society India. Microbiology Society India, it was established in 1996 under the leadership of Dr. Arvind Deshmukh. Sir, at that time, very few members were there, mostly the 10 members were there initially, and now more than 1,000 live members are there on the roll. Apart from that, they from India and abroad also. We are much more student centric. We are conducting many more activities for students, even for the researchers and also for the industry people. Students, more than 20,000 students, they are associated with the Microbiology Society India across the country through different student units and from the Telegram groups. So we encourage particularly the liaison between government and non-government organizations for the different problems and giving the different solutions for the problems in the field of microbiology, which are much more related to the society and in the academics or in the research. So also we encourage the organization of different type of the conferences, seminars, then the webinars and so on. And also we are giving the career counseling type of the services to the students, particularly in the field of academics, in the industry, and so on. So entirely we are going for giving the different, we are tackling the different type of the things related to the student, society, research, and so on, related to microbiology and biotechnology. So you are welcome, sir, in
Thank you,
this
sir.
platform.
Thank you, sir.
Thank you, Sanjeev, sir. So now over to Dr. Nagesh Kadam for his talk, and it's on a very important topic. As per my knowledge, C. elegans is a type of nematode which is generally non-parasitic in nature and is harmful to humans. But due to its like very simple genetical framework, it generally has a mimicking power to the human genome. So it can be used as a model for various type of disease studying model, as a disease studying model. So over to you, sir, for enlightening us with this particular topic and a very important aspect. Over to you, Nagesh, sir.
Thank you. Thank you very much for the kind introduction. In this talk, can you see my presentation now?
Yes, sir. Yes, sir. It's visible.
Okay. Thank you. Okay. So in this talk, I will introduce about the C ligands. What are the C ligands and what is their role to understand the different biological questions. So it's going to be very general topic. I will introduce about some aspects of the C ligands used to ask the questions in the developmental biology field, in the aging field, or in the nervous system, or the people, those who are working on the neuroscience field. So coming to the C ligand stands for Synaptic elegans. So the talk will go through like the introduction and what is the use of C ligands in the developmental biology, in aging, in our system analysis and functions, and then for the conclusion. So coming to the model system, because most of the times I'm telling you that this is a model system. So to be a model system in the biological field, the organism or any bacteria or any multicellular organism should have some characteristic. It should be inexpensive to maintain. The organism should have some short lifespan, so you can trace what is going on with the animal in a short duration time. The organism should be genetically manipulable. Means that you can trace the genes and you can manipulate the genes, and you can change the protein level and other things, and you can track down what is going on with the DNA regulation and transcription, translation, etc. And it should be promised some biologically important questions. So with this, there are several model system has been introduced in the biological field to understand different questions like bacteria, yeast, cell culture, there is a nematode, there is a fruit fly, mice. So depending on the questions, people use different model system, like for gene expression and some protein expression, mostly the people use bacteria and yeast. And for the nervous system and developmental studies, people use nematode, fruit fly, Drosophila as a model system. See, so in this talk, I will talk about nematode, which can be used as a model system, not only for the nervous system function, but to look at different questions in the biological aspect. So C. elegans is a Synaptic elegans, as I said earlier. It's come under the phylum Nematoda. It's a microscopic worm. So this is like a C. elegans. It's a microscopic. In actual reality, in the lab, they are grown in the plate where they feed on the bacteria. These are like in nature they could be found in the moist soil, and naturally they eat on the bacteria. So they don't need any complex things to grow them. So it's very simple thing. We just need to plate the bacteria on the plate, and these worms, C. elegans, they feed on those bacteria and they grow. And they have this simple anatomy. Anatomy means like if you look at this bacteria here, this is like, sorry, can I use the cursor also? Okay, here these are the C. elegans in the bacteria. They are eating the E. coli. Here this is like a 0.5x magnification. You can see in a single plate you can grow like thousands of worms. And if you go close at 10x objective, you can see these are like adult worms. They have different larvae stages, and these are the eggs laid by this C. elegans. And they have this simple anatomy structure, like they have this head region, which we called pharynx. is attached with the intestine, where they ingested the food, and they have the gonad and muscles and the nervous system. So this is like a simple body structures they have. They are not complex like us. And they have simple behavior, like this is the sinusoidal movement. They just crawl around the surface. That's their simple behavior. So, but this system is used for several biological aspects. And apart from that, these animals also has short lifespan, means that from the embryonic stage to becomes reproductive adult, it takes only 2 to 3 days. And it goes in the aging process to becomes naturally dead. It takes around 2 to 3 weeks. So it's like within 1 month, the full life span of the C. elegans is complete. And within 2 to 3 days from eggs to become adult, they becomes like take this short duration time. And apart from that, these C. elegans, they are hermaphrodite. Hermaphrodite means that they have this female and male sex organs. They are within the same animal. So in naturally, there are 2 sexes are present for the C. elegans. One is hermaphrodite, which has male and female sex organ in the same animal, and there is a male. So these are the 2 sexes are present naturally, and the natural so natural frequency of the male worms is very low. Naturally 0.5% in the population, they are male. So as such, C. elegans don't need male worms, and without male fertilizing the C. elegans, this hermaphrodite can produce around 2 to 300 progeny worms. And if they copulate with the male worms, they can increase the their progeny to 1,000. So these animals, they can, like bacterial colonies, they can self-propagate. And if you mutate any gene in the C. elegans, it will self-propagate to the progeny, and you can have multiple copies of the same genes or same mutation in the whole population. So just by maintaining the one C. elegans and their progeny, we can get like multi-copy progeny of the same gene. So apart from that, they have this transparent body. So here, these are some examples. Here people have used different promoters to express some set of fluorescent marker genes in the neurons, in intestine, in some specific set of neurons like GABAergic neurons, glutamatergic neurons, cholinergic neurons, some muscles, some germ lines, epidermis. So the genome sequence is completely sequenced for the C. elegans, and we know exactly which gene is residing on which chromosome and what is their sequence. So on the basis of that, people have used C. elegans extensively to study the role of different genes, and through that they find out this kind of different genes they are expressing in specific tissue or specific cells. So on the basis of that, they have they have expressed the promoter with some fluorescent marker like M-cherry or GFP, and you can tag by using that specific promoter, you can tag that specific set of genes, like here is a cholinergic neurons. So you can trace what is going on in the cholinergic neurons by tagging the promoter with GFP. And so due to this simplicity, they can be characterized genetically. And if you see the gene structure and gene composition, about 70% of the human genes have related genes in the C. elegans. And with this animal was firstly introduced by Sidney Brenner in 1964, but he's also well known for his discovery of the messenger RNA, and he also helped to solve the genetic code. But once during this golden era of molecular biology, when people have discovered the DNA and the genetic information and the codon and everything, he wanted to study the nervous system. For that, he looking for some simple model system to study the neurons and other things. So for that, he introduced C. elegans as a model system for the nervous system study. And during 2002, he shared a Nobel Prize with Robert Horwitz and John Sultan for their discovery and concerning the gene regulation of organ development and programmed cell death. So C. elegans was the first animal where the apoptosis and the RNA interference was firstly discovered. So due to their discovery, they have shared the Nobel Prize. And he also published one paper, which is like a hallmark for the field, which is like a genetics of C. elegans, which was published in 1974. Here, he mutagenized C. elegans. Hello.
I request Dharendra Kumar to please mute yourself.
Okay, so he made some random mutation in the C. elegans, and he found the genes causing different phenotype in the C. elegans. Like some animals become smaller due to set of gene mutation. Some animals becomes dumpy; they are thin and smaller. Some animals shows egg-laying defective phenotype. There is no proper development of the vulva, through which the worms lay eggs. And some animals has this protruding vulva mutant phenotype, which is like the vulva is like outside instead of inside here in the C. elegans. So this kind of visible phenotypes they see due to the mutation in some set of or single gene. So these are like still people are using this mutation for their studies and to look at the pathways those who are regulating this path, this behavior, this phenotype. Sorry, there is something wrong. Okay, sorry. So for to mutagenize C. elegans, the classical technique is to use some chemical agent is called ethyl methane sulfonate, which is like a chemical mutagenic agent. We just need to exposure the C. elegans with these chemicals. Then it causes the random mutagenesis in the C. elegans genome. And people then screen the progeny of those worms, because you need to just exposure the worms to the mutagenic agents, then single out those exposed worms to these agent, which causes random mutagenesis in the C. elegans. And once the parent worm like F1 worms start giving the progeny, it will give the progeny of their own gene duplication. So you will have the lot of worms from the single animal. So on the basis of if there is a mutation causing some kind of rare mutant phenotype, then you can isolate those worms, look for their you can do the genome sequencing and other thing like the chromosome mapping, and try to find out the new genes. Like that, several labs all around the world find out different genes and functions of the different genes in regulating different whatever I showed you earlier, different phenotype, different behavior, other things. So this is like a very classical mutagen agent which is used to mutagenize the C. elegans genome. But recently people have also started using this transposon-based mutation, which is like a very directed mutation. If you want to mutate any single gene, you can use this transposon-based mutation. There is also people use RNAi, which is like RNA interference, which can be just you need to feed the C. elegans with this RNA expressing bacteria, because C. elegans eat bacteria. So just if you need to feed the worms with the double stranded RNAi, so the targeted genes will get knocked down. So this is like one standard techniques people are using to screen multiple mutants. And recently, with the revolutionized CRISPR technique, people are using extensively this CRISPR-based mutagenesis techniques, where they can specifically target the gene inside the chromosome, and they can tag and delete and mutate like single point mutation in the C. elegans genome or in that specific gene, and try to find out the role of that gene in whatever they are looking at, like the phenotype or the behavior, like that, or developmental aspect. So coming to the developmental aspect, so as I showed you earlier, so this is the C. elegans, this is the gonad. They form the oocyte. As I told you, the C. elegans, they are hermaphrodite. So they have this spermatheca within their body. Once the oocyte comes near to the spermatheca, they get fertilized and they form the embryo here. So, and C. elegans, they are transparent. So you can see the direct cell division in the C. elegans, and people are extensively using this model system because there are, like, you can grow them in a small plates, and you can grow them in a very fast manner and in a huge number. So people are using this as a model system to study the genes those are involved in early cell division and embryogenesis. So this is like you can trace down by tagging the gene with mCherry or GFP or any fluorescent marker. So here is one example. People here, Julie Arranger lab, they have used, they have looked at some molecule that is a TAC1, which they have done the RNA. Like as I showed, you can suppress the gene functions by just feeding the worms with the RNA plasmid. So this sorry.
So please continue. I think it was just like.
Okay. So this protein has functions which stabilize the microtubule growth regulator. It's act as a microtubule growth regulator. So here, this is like a wild type. This is like this is the male. Here they are fusing, the sperm is fusing with the oocyte.
Capital M.
So then the two nucleus, they are fusing and they start like making a two cell stage, and others starting the embryonic stages. And here you can see in the wild type there is a normal first division, but if you mutate this specific gene through RNAi, there is an inhibition of formation of this cell division pattern, and most of the worms, most of the embryos, they are dead. They are not developing properly. So this is just one example, but people have used to look at the morphogenic gradient in the developing embryo, how the cells they are deciding which end to go towards the anterior and which tail go to the posterior position. And there are some other, like several P granules, and there are several other things in the during the embryogenesis, how these are regulated and how the embryo is forming, and what are the like molecules involved in regulating these genes, and the morphogenes regulating this axis formation of the anterior, posterior, ventral, dorsal, and organs during the early embryonic stages. There are several studies they are going on. So this is just one example. And here is another example. The C. elegans, this is like a first multicellular organism which genome is sequenced. And we also know that from the zygotic 2-cell stage, which get divided into like 8, 16, 32 cell stage, they have known that the particular pattern of cell division for each and every cell. So from this each and every cell, like what they are going to become in the adult, they call it as cell lineage pattern. So from the early embryonic stage, you can trace down on the position of that cell what it going to becomes in the adult. Like some set of cells, they become hypodermis and neurons. Some set of genes cells, they become neurons and vulva in the adult worm. Some they become muscles. Some cells, they become somatic gonad. So, and this whatever the cell lineage pattern is constant all over the C. elegans. Like it will not change at any time. So this is like a constant cell lineage pattern. They have this constant fate. So people are using this information to trace how this constant, like the polarity of the cells and the destination and the latency, how their fate is determined, what are the factors, those who are determining their fate from the early embryonic stage. like morphogen gradient, there are some heterochronic genes. There are several other proteins also. They are involved in maintaining this kind of pattern in the C. elegans lineage development. And here is one example. Here we see this is like a wild type worm. If you mutate this RAS, which is like GTPase, one of the GTPase, which is involved in some cell fate transition. So the level, when there is a gain of function mutant, you can see instead of one vulva here in the wild type, in the mutant worms, we see this multi-vulva phenotype, because these genes, they are involved in making the vulva during the cell fate transition. So this is like act as a switch between vulva and hypodermal cell fate during C. elegans vulval induction. So just due to the gain of function of this specific gene, there is this kind of phenotype in the C. elegans. And if you here, I have not shown here, but if you mutate this gene, there is a vulvaless phenotype. There is no vulva formation in the C. elegans. So, and if you overexpress it, then there is a multi-vulva formation. So kind of these GTPases, we don't know exactly how many genes they are regulating, but it's regulating the vulva formation induction cell fates. So these kind of genes have been studied by several labs, and still they are studying how the different cell types get their fate throughout their development, and which are the molecules they are regulating, and they are making their fate to becomes like hypodermis or neurons or vulva. So this is like studies, still people are using it, and they are looking at some transcription, translation, protein expression and gene expression profile in all these cells to look at how does this fate transition happen by using C. elegans as a model system. And coming to the neurons, because initially it was proposed to be look for the nervous system. In case of humans, we have these billions of neurons, and they make like trillions of synapses, and it's very hard to study the single neurons as such in the C. elegans. That's why we need to use some simpler model system. People are using the neurons cell culture, but we need some like animal model system for that. This C. elegans is a very useful tool to look at the neurons. And one of the good thing about C. elegans, it's connectome. Connectome is like genome sequencing, where we know the gene location and their location.
और दोनों कम में मैं मम्मी को 2-3 ऑप्शन दिखा सकूं जरा.
So in case of C. elegans, the connectome, like the neuronal physical map, is completely known. People have traced down each and every neuron in the C. elegans body by using electromicrograph technique. Like it took like a lot of time, but people have identified the neurons, their connections, the type of neuron, and the neurons where they are forming the synapse on the C. elegans body. So structurally, it's well known that which kind of neurons they are present at which location, and they form synapse with which kind of other neurons. So this is like going to be a first multicellular organism where their neuronal map is completely known, and they just have only 302 neurons. So it's easy to look at the neurons instead of like mice or Drosophila, which have thousands and billions of neurons. And through that, we can study the functions of the neurons in different behavior, because C. elegans, as I showed you, just it's crawl, but it's in later I will show you which kind of behaviors they perform. They also try to find out the functions of these different receptors. Those are present on the neurons, and sometimes some set of neurons, they get activated in certain specific behavior. Like there are some behaviors which activated by people have identified this set of neurons, they are getting activated, not by all the neurons. So through this C. elegans, we know there are how the neurons they are like working in some specific set of behavior. So as I showed you earlier, this worm shows this crawling behavior like the sinusoidal body movement, but this body movement is also controlled by different neurons. So here is one example. There are some cholinergic neurons and GABAergic neurons, which releases acetyl. The cholinergic neurons releases acetylcholine on the muscles, and the GABAergic neurons releases GABA on the muscles. So whenever there is a release of acetylcholine on the muscles shows the contraction, and whenever there is a release of GABA, GABA butyric acid, then the muscles shows the relaxation. So these motor neurons, they act in a, like they fire in a specific manner that the worm shows this kind of behavior, where you can see there is a ventral relaxation and dorsal contraction and ventral relaxation. So through that, the elegans maintain their this sinusoidal movement, locomotion. And by using some drug molecule, you can trace down what are the molecules or receptors they are present on the synaptic junction, like on the muscles, or what are the molecules they are present to release these vesicles from the presynaptic neuron. So for that, there are some different drugs which can be used. And also this locomotion is also controlled by some sensory neurons, because elegans has 302 neurons, as I showed earlier here. So most of the sensory neurons, they are present in the head region and the tail region. And these sensory neurons, they also senses the surrounding stimulus around them. Like they can be chemosensory neuron. They have this different kind of sensory neurons. One set of neurons, they sense for the chemicals in their surrounding. Some set of neurons, they sense for the mechanical stimulus. Some neurons, they sense the temperature gradient around surrounding their environment. And some neurons, they also sense the taste. So these neurons, once these neurons get activated by different stimuli surrounding them, then they activate these interneurons. Interneurons, they kind of like decision-making cells here. And these interneurons then act on the cholinergic or GABAergic motor neurons, which causes muscles to get relaxed or contract. And on the basis of these chemical cues, worms will decide whether it should go forward direction or go to backward direction and run away from the stimulus. So this is just a simple behavior, but it becomes complicated when you go to at the neuronal level, because there are different set of neurons. They get activated. They give signal to the other neurons. These other neurons give signals to the motor neurons. Motor neurons then act on the muscles, and then the actions. Then the action is taken by the elegans, whether to go in the forward direction or to the reverse direction. So here is one example how to decipher the molecules those are involved at the neuromuscular junction. So the molecules, the worm show contraction and relaxation. There is one drug called aldicarb. So this is like aldicarb assay in wild type case. So aldicarb, normally when there is a release of acetylcholine in the synaptic cleft, it binds on the acetylcholine receptor and it triggers the muscles contraction by increasing the calcium flux. And whatever the acetylcholine present in the synaptic cleft is degraded by cholinesterase enzyme. But this drug causes acetylcholinesterase enzymes to become inactive. It blocks its activity. So there is a rapid influx of the calcium, which causes worms to paralyze, because there is continuous contraction in the muscles and the worms will not move properly. So there is a paralysis of the worms due to this aldicarb drug. And this drug can be used to trace down the molecules which are regulating the receptor number here, or the release of acetylcholine from the synaptic cleft. So there are RIC mutant and HIC mutant. The RIC are the resistant to the aldicap assay. So here, this is like wild type line, and RIC is like this. So the wild type worms, they get paralyzed around in suppose 100 minutes. And the RIC, those who are resistant to the aldicap, means that they have lower release of this acetylcholine here. Then these animals shows there is a increased resistance to the paralysis, like towards the aldicap. So, and there are some mutants which releases too much acetylcholine in the synaptic cleft, which shows compared to the wild type worms here, they shows earlier paralysis in presence of aldicap. So on the basis of this assay, you can find out the molecules those are involved in synaptic cleft on the muscles, like the postsynaptic site or the presynaptic neuron. So on the muscles, these proteins, they might be cell adhesion molecules or some other molecules, those who are regulating the receptor numbers, those the receptors, those who are responding to the acetylcholine, or maybe some presynaptic molecules, those who are releasing too much acetylcholine from the presynaptic neuron, and which causes rapid paralysis. So on the basis of that, you can trace down the role of any specific receptors. Those function is unknown in the cell in the synaptic cleft. So here is one example. It was reported by my supervisor when I was in PhD. So in wild type worms, this wild type worms shows paralysis in 20 minutes of time. But when they have mutated one molecule, which is like a cell adhesion molecule in the synaptic cleft called RIC3, these animals shows rapid paralysis rate compared to the wild type. So this is like this molecule. Further, they have found that this molecule is regulating the CR16. That like this is the receptor number on the muscle cell. When there is a mutation, there is too much acetylcholine receptor on the muscles, which causes rapid paralysis of the worms. And when there is a presence of this actual RIC3 molecule on the membrane of the muscles, neuromuscular junction, they shows like normal paralysis. So like this, there are several different molecules and presynaptic molecules. They have been identified through this simple behavioral assay. So these worms show paralysis and not paralysis. So on the basis of that, we can find out new molecules. And here is another example how some set of neurons activations can trigger the specific behavior in the C. elegans. So this is like a mechanosensation in C. elegans. It is sensed by these three sets of neurons. One is ALM. This is like a neuron cell body, and this is a anterior process. This is the AVM neuron. It has it's beneath the C. elegans, so you can't see the process of the AVM neurons, but it also going along with the ALM. And here is a PLM neuron. There is a PBM neuron under it. So these are the neurons. They supposed to sense the gentle touch to the C. elegans. And in one of the report, they have found that there are set of neurons. They get activated when there is a mechanical stimulus to the head region, like these ALM and AVM neurons. They get activated. They further activate some set of neurons in the head region, so the head start moving back, and then it turn completely head to the back, and the worms move away from the stimulus. So like this here, when there is a mechanical stimulus, worm is like moving back. So there are set of neurons, they get activated, and then further they activate this like muscles. Like these are the like motor neurons. They act on the muscles, like in the dorsal muscle and ventral muscles. They act on these muscles. So there are set of different neurons. They get activated for each and every behavior the worm shows. So like this, there are several in the research field. There are several neural circuitry have been deciphered. which regulate this kind of different behavior in the C. elegans. And on the basis of here, they have looked in this paper, they have looked at this tyramine, which is like one of the neuropeptide, which act in the muscles. And if there is a mutation in the tyramine gene, then the worms will not show this backward head movement. And there is a defect in the this backward movement of the worms. So through these different molecules in the nervous system has been deciphered, and still they are deciphering. And how does this different networks they modulated in different behavior is still a question, because this is like a simple behavior, but you can see here there are different set of neurons. They get activated, deactivated, and there are some activator and activator and terminator for these different sets of neurons. So to understand this complete set of circuitry under some simple behavior of the C. elegans, it's like it will take a huge time. So still there are several groups they are trying to identify this different behavior and the networks, neuronal networks behind those behavior, and the molecules those are involved in the behavior. And here is one example by the simplicity of C. elegans. Also, you can trace what is going on inside the neuron. So here is a one movie which I have made. I am working on these two neurons also. So this is like a PLM neuron. Here, this is a PLM neuron. Here, the neuron is the neuron. As I think we all know, there are some synaptic vesicles inside the neurons. They transfer the different chemicals to the postsynaptic and presynaptic terminal. So here is the one movie. It's okay. Okay. So, this is a movie. Oh, sorry. Okay. So here these are so in the live animals you can track the vesicular movement inside the neuron. So whether due to some mutation, how the vesicles they are getting trafficked inside the neurons, their run length, their velocity, their speed, everything can be calculated by using C. elegans as a model system, and that is in live animal. So this is like a beauty of C. elegans. You can use it from tracing the neurons, and also you can look the calcium dynamics and what is going on inside the neuron. Apart from that, C. elegans is widely used for the aging studies. Like the humans, C. elegans also shows this aging, and during aging there is a degradation of the muscles, the fragmentation of the muscles. And there is a neurons, they show debranching, and there are so many like neurodegeneration kind of phenotype, and the reproductive system becomes less active. So this is like whatever we see in humans, it's similar with the C. elegans as well with the aging aspect. And there are several molecules has been studied and has been known now that increase the longevity of the C. elegans. The molecules those are involved in the insulin signaling pathway. They are also involved in the germline stem cells. Some molecules they shows the increased longevity by doing the dietary restriction in the C. elegans, and also through the mitochondrial impairment. And these all are regulated by the epigenetic alteration as well. So there are different molecules has been studied in C. elegans which regulate the longevity or the aging of the C. elegans. So here is one example. This is like a graph which shows the survival rate of the C. elegans. And here this rate, just a minute. This white line shows the wild type worms. They become dead in around 20 to 30 days. And if you mutate this one gene, which is a DAP2, which is involved in the insulin like signaling pathway, the worms, which is living around 20 to 30 days, the lifespan become increased double to become 50 to 60 days. So, and also if you mutate this EAT2, which is involved in the dietary restrictions, like EAT2 mutants has some mutation in the pharynx, in the mouth region of the C. elegans, where they can't eat properly, due to which they go on the dietary restrictions. And due to the dietary restriction as well, there is an increase in the lifespan of the C. elegans. Like instead of 30 days, they almost live around 40 to 45 days. And these two pathway, like they are kind of combined in a such a way, because the beauty of C. elegans again is to make, you can make the double or triple mutant and to trace whether these two genes, different genes, they are involved in the same pathway or not. So here you can see like the double mutant of EAT2 and DAP2, they almost showing a similar phenotype. Yeah, they are not showing the similar phenotype, suggesting that they are mostly involved in the similar kind of pathway, or maybe some yeah, by maybe regulated by different signaling pathways inside the C. elegans, but somehow they are increasing the lifespan from 30 days to like 60 to 70 days. So there are, and people are still studying, like there are not just these two pathways, there are multiple pathways which regulated through these signaling molecules, and people are studying how, what is like their role in such as in aging. So C. elegans has been a good model system to study the aging process as well. So with this, so C. elegans is like an elegant model for different aspect of the biology to study like anything from the development to the aging. But whatever I showed you just is a spoon of information in the ocean, because people have been using C. elegans as a model system to study like epigenetic information, how the information is going from parents to the progeny. There have been some labs, they are doing this interesting stuff. And also people are looking at how does the C. elegans pass their memory, and how the memory in C. elegans is formed, and whether the C. elegans can remember something or not. So people are also working on this memory aspect. There are some labs, they are working on the evolution by using C. elegans as a model system, like evolution in the genome and other things. And also for microbiology society, I guess there are some labs, they are working on the intestine of the C. elegans. They feeding the C. elegans different bacteria, and they are looking at the aging aspect and also the neurodegeneration and other things. So yeah, it's quite a good model system to study different aspects related with the biology. So that's it. Thank you very much. Thanks. This is my lab.
Thank you so much, sir. This was an amazing session and a very informative session as well. And sir, we'll be proceeding towards some questions and answers. If somebody has some many queries regarding C. elegans, you can just write in the chat box, or you can just unmute yourself and ask. Anyone?
Hello, good afternoon, sir.
Yeah, good afternoon.
Yes, sir. My name is Juhi Goyal, and I'm a PhD scholar in the lab of neurobiology at Mohanlal Sukhari University, Rajasthan. So I wanted to ask whether, if suppose I want to develop the C. elegans culture in my lab, so do I need any ethical clearance for that?
No, I don't think C. elegans come under any ethical committee, because they don't sense the pain as such. So I don't think so you need any ethical committee. But exactly, I don't know that much, but you can ask to some, maybe some HR in your university, or some like people, those who are in the ethical committee board.
No, no, they are not. It is not required, I think.
Yeah, I don't think so, because we never had any like kind of committee in Hyzer Mohali.
Ethical committee, it is only for animals, particularly mammals and other things. It is required.
Okay. Thank you so much, sir.
There are so many references for the RNAi
Yes,
study.
sir. Yeah.
Lot many work it has been done on the RNAi in the C. elegans.
No, sir, there are several work has been done. Like people are still using it.
Yeah.
If suppose they want to, because it's not like permanent deletion or termination of the gene, it's just a knockdown.
Yeah.
So it's just live like for 1 or 2 generation of the C. elegans. That's it. So whenever people are looking for some phenotype, they are soaking the worms, just feeding the bacteria. Those are expressing this.
E. coli particularly.
E. coli, yeah.
Yeah.
And they are like knock down the expression of that particular gene.
Anyone with the additional queries?
Hello.
Hello, sir.
Yes, sir.
Am I audible to you, sir?
Yes,
Yeah,
sir,
yeah,
you are
yes.
audible.
Sir, I would like to know that there are some fluorescent tagged C. elegans.
Yes, sir.
If you want to use them for the study of some antimicrobial effect or some physiological effect on them, so what will be the parameters so that we can choose that this type of mutant will help to study? Like we want to test the toxicity of some antimicrobial compound
Okay.
on some fluorescent mutant of C. elegans. So which strain will be suitable for that?
Sir, there are some strains which have this superoxide dismutase and other molecules which are tagged with some GFP, I guess. Those can be useful for this kind of studies. Maybe like depending on what you are looking at, there are like several lines available like in the C. elegans database and on the labs. You can just request them to have your lines. Like if you're looking at some set of genes or some set of molecules, you can also get those lines.
Okay.
So
we have different mutant for different function in GFP tagged.
Yes, sir. Yeah, actually, you can make because making a mutant in like if the mutant is available and it's not available in your desired GFP or MCD background in the transgenic strain, you can cross that strain with your mutant and see what is going on, what is like phenotype in your mutant case and in the wild type case with the GFP or MCD tag. So you can like cross fertilize the C. elegans, like just Mendelian cross. You can get whatever you want in the like genetic background if the mutant line is available.
If you want to procure some strains in India, some strains of C. elegans we want to procure because we don't have the facility or the strains we have. So from where we can get it, sir, easily?
So you just need to request some labs. There are some labs in India, like there are now I think 7-8 labs in the India, like in ISC there are 2-3 labs. In TIFR there is one lab. In ISER Mohali there is one lab. Like my lab was there before. Now it's in ISC. Yeah, but you can like there are several labs in the India. Just you need to ask them, request them to send the C. elegans. I think because when I was in India, we were sending like wild type C. elegans to the different labs, different universities whenever we are getting the request. So I think the community is helpful. You just need to ask them and what you want to do with those ones. So I think that's it. You just need to request them.
Okay, thank you so much. Thank you so much for your valuable suggestions and answers. Thank you so much, sir. Thank you so much.
Thank you.
Any more queries? So, sir, I have a curiosity, like more than a query, that we are using C. elegans as a model for studying various anti-aging processes. So how will you rate the success? Like what will be the success rate at present for using it as a model?
Success rate means what?
Means
like
how much it matches with the like expectancy, like we are going for a particular process reading of like anti-aging processes and all. So how would you rate it as a model? Like, do we have any obstacles still left, like to overcome and all?
No, obstacle in the sense of what? Comparing C. elegans with some higher model system?
Yeah.
Actually, the molecular pathways and the molecules, they are highly preserved. I don't think that like there are as such obstacle like the DAP2, one of the enzyme which regulating this insulin signaling pathway, which has been reported in dog as well. It increases the lifespan of the dog. So I think it's very like preserved everywhere in the animal phylum. So maybe you can, if you are getting something, then you can try with some higher model system just to confirm it, like with the mice or something. But I don't think that is useful that much, because people are considering C. elegans as a robust model system for the aging studies.
Thank you so much, sir.
Thank
So
you.
if anybody has any query, or I should now call upon Sanjeev sir for the vote of thanks finally now. Over to you.
Before concluding, please, may I know Dr. DC Sharma, sir, from where you are?
Hello, sir.
Hello.
I am Dr. Deepak Chand Sharma from Lucknow, sir.
Okay.
I am assistant professor in Department of Microbiology. Dr.
Yeah, that's that I want to know. Yeah.
Okay. Sir, I think in Lucknow also there is one lab.
Yeah, Dr. Rakesh Pandey is there in CMAP.
Yeah, I think in CDRI also there is one lab. Amir Nazir, I guess.
I have no idea about that, but Dr. Rakesh Pandey, we have good communications, and we got mutant C. elegans, wild type of C. elegans from there.
Okay.
And we are working on that. But we come to know that some green fluorescent protein tagged C. elegans are available. And if we work on them, some tracking of gene is very easy.
Yes. It's very easy.
Basically, I'm a microbiologist, so I would like to know more about the C. elegans.
Yeah.
It is a good model organism to work.
Yes.
Okay. Nice to see you, Sharma.
Thank
you. I am very much thankful to Dr. Nagesh Kadam for joining this session and enlightening us on the C. elegans as a model for the different type of the studies. I am also thankful to participants, those who have joined and shown their interest in this particular session, that is Indian speaking from abroad. Also, Nitika Thakur, she also has conducted it in a very nice manner. So with this, we conclude the session. So thank you one and all for joining, and we expect in the future also, Kadam sir, your cooperation and the certain type of the inputs regarding the different things. And also, I wish that in the future lecture series, more participants should come and take the advantage, particularly the MSc and the PhD students. So thank you once again, you all.
Thank you. Thank you very much for having me here.
Yeah.
Thank you so much. Thank you so much.
Thank you.
Thank you, everyone. So take care and keep growing and keep glowing. And we'll be meeting in the next series of Indian speaking from abroad. So this Sunday, we'll be meeting again. So take care and bye-bye.